Okay. Very good evening and contact to all this webinar. Nice topic of bacterial production and Type VI Secretion system is delivered in a therefore colleagues Department of Microbiology available library and grateful for the management of treatment as is still
Thank you, Sujal Pal. It's my great pleasure to introduce Dr. Vishwana Jana, Postdoctoral Fellow, Department of Clinical Microbiology and Immunology, Tel Aviv University, Israel. Dr. Jana has completed his BSc in Chemistry and MSc in Genetics from Calcutta University. He did his PhD under the guidance of Professor Subroto Shao in the Department of Biochemistry at Bose Institute, India. His PhD work focused on biophysical and biochemical characterization of a MIP-like protein from E. coli. After his PhD, he joined the lab of Professor Indranil বিশ্বাস as a postdoctoral fellow at the University of Kansas Medical Center, Kansas, USA. In his postdoctoral study, he worked on the functional mechanism of CLP gene in Streptococcus mutant. In the present lab, Dr. Jana is working on the T6SS as a new antibacterial platform. Dr. Jana has published several research articles in high impact journals like Nature Communication, Biochemistry, Life Science Alliance, and Ansphere. Now I am requesting Dr. Jana to deliver his lecture on the topics bacterial warfare mediated by type VI secretion system. Dr. Jana, please.
Thank you very much for the kind introduction, Subroto, and thank you very much for the Ramkishan for giving me the opportunity to present my work in front of the wonderful audience, which is young mind, curious mind. Hope you guys will enjoy my lecture. Can you see my PowerPoint?
Yes, sir. Yes, sir.
Yes, sir.
Okay. Thank you. Thank you. Okay. As Subroto said, today I will present my work, which I'm working specifically in this lab in Tel Aviv University. Our lab is mainly working on bacteria, so we are mainly working on bacteriology and bacterial genetics. And mainly, as the title suggested, that our lab is mainly working on bacterial warfare, that bacterial fight they do each other. Okay, so let's start. As you all know, bacteria are found everywhere. Like if you can see, the bacteria can be found in the pool that is found in Yellowstone. So this temperature has almost 70 to 80 degrees, but bacteria still lives there. And as you can see in the deep sea ocean, in the deep of the ocean, the bacteria lives there. As we all know, bacteria is also found in our human body. And basically, in human body, almost the same number of bacterial cells are present as like the human cell. Almost, I found a few years back, there's a report that almost 10 to the power 13 bacterial cell lives in a 70 kg human. So, but this is the bacteria is very small compared to eukaryotic cell. So, its complete weight is only 200 grams. But if you count the number, is almost the same as the human cell. So bacteria is a social organism. It's not like that one bacteria lives here, another bacteria lives here. So they live together. So they continuously interact each other. So this interaction sometimes could be they use for the beneficial for that. So they help each other to grow. Like in the quorum sensing, they can talk to each other. They interact with each other like this by using some signal. So they secrete some synthesis. They use some synthesis gene to secrete some signal molecule, which the signal is recognized by neighboring bacteria, by same group of bacteria by the receptor. So they can communicate each other using the secretion signal. So when to they form biofilm, when to attack, when to secrete the virulent toxins, they use this signal. But this coalition is always not peaceful. Sometimes they also fight to each other. Okay, because of the space and nutrients, and sometimes against the host immune system also. Okay, today basically I will discuss only for not about that fight against the host immune system, but today I will discuss mainly the fight between bacteria against bacteria, what the different weapons they use. So they use a varieties of weapons to fight each other. Like one is called the bactericin. What is bactericin? The bacteria secretes some large toxin molecule from bacteria, and which somehow can kill the neighboring bacteria, which is not safe. And they also use some another system like the bacterial protein secretion system, varieties of protein secretion system. So depending upon the protein which is involved in the secretion, it can be divided from type 1 to type 9. One second, can you see my mouse, please?
Yes, we can see it.
Okay, thank you. Because I use Zoom platform, so Zoom platform there's something else. Okay, thank you. So depends from type 1 to type 9 secretion system, depend upon the which proteins are involved. So they are completely independent of each other. It's not like the certain set of protein is like from type 3, that's a different set of protein from type 4. So depend upon the mechanism, the secretion system can be divided in two ways. One is one-step process. In one-step process, this molecule directly from the cytoplasm of the one bacterium is delivered to the neighboring cell, directly into that, inside there. As you can see here, type 3, type 4, and type 6. Okay, but there is a two-step process also. So there, the molecule is first transferred to the periplasmic place of the bacteria. Then from the periplasmic place is delivered for the outside. Okay. So they deliver the different varieties of molecule, like type 4 secretion system. They mainly deliver the molecules is DNA. As you know, the conjugation, in conjugation, basically type 4 secretion system plays a major role. So use the DNA from one bacteria to the another bacteria. Like type 3 secretion system, also it can mainly deliver toxin in the eukaryotic cell, not in mostly in eukaryotic cell. So it mainly delivers that toxin, which is virulence toxin. Basically, it's not good for the eukaryotic cell. Today, I will discuss mainly type 6 secretion system, because its interesting thing is that about type 6 is that it delivers the toxin into neighboring bacterial cell. So mainly target bacteria. So it plays a major role in interbacterial competition. So to fight bacteria, bacteria use this type 6 secretion system. Okay. So if you see that basically type 6 secretion system as the ancestor from T4 bacteriophage. This is the T4 bacteriophage. Okay, so there is if you consider this protein like this protein is almost the same as the outer protein of the type 6. So it depending on the resemblance, so we found that type 6 secretion system is mainly come from as the ancestor from T4 bacteriophage. Okay, so but with during the horizontal DNA transfer, bacteria took this DNA and they modify it as a protein secretion system. What is how it works? Basically, it works like this. So it's first from the sheath protein, and then this is the toxin, which is mainly loaded at the sequence system. It acts as a bow and arrow. So let's say it's an arrow, and this is the bow, and then this arrow goes down, and then this deliver the toxin into neighboring cell, and this toxin is mediated the toxin activity. So it basically system is a contact dependent toxin protein delivery apparatus. Is only found in gram negative, not in gram positive. This particular type C secretion system. And it's only found not all gram negative bacteria has type 6, only 25% of gram negative bacteria has a type 6. And it plays a major role in interbacterial competition. As you can see, it can deliver toxin to the neighboring bacteria. So neighboring bacteria can kill or compete with the bacteria. And the toxic activity is mainly carried out by the toxin. It's that punching don't harm anything. Only if it can deliver the toxin, then it kills. So I'll show you some structural dynamics, which is published just few months back in a review article. So first, they form in the membrane, they form this base plate, and then all this complex protein is found. And this is the outer membrane, and this is the outer sheath protein, and this is the inner sheath protein. Then it's deliver the toxin to the neighboring things. Okay, I told you earlier that this toxic activity mainly carried out by different toxin to that. But what's the target? How is this toxin kills neighboring bacteria? This toxin basically target the essential component of the neighboring bacteria, like the peptidoglycan hydrolase, as this is found in inner outer membrane. So if you degrade peptidoglycan hydrolase, so cell burst basically. Also, it can make a pore forming. It make a pore into the membrane, so bacteria die. And then it can also target the inner membrane by targeting the lipase. So it can degrade the fatty acid. At the same time, it can target some essential protein of the bacteria like FTSZ, which is mainly responsible for bacterial cell division. So if it can target FTSZ, so if FTSZ cannot work, so bacteria also die, and also target genomic DNA, like it can also have DNase activity. So it can target that and the genome of the neighboring bacteria or competing bacteria. So you target the DNA, so neighboring bacteria will die also. So it has varieties of activity. So it mainly target the competing bacteria. Now the question is that it delivers the toxin to the neighboring bacteria and kills. Then, but the toxin is produced inside the bacterium. But how this bacterium can prevent itself from its intoxication? How can bacteria protect itself from this own toxin? So in that case, the bacteria basically is a toxin. Basically, the toxin which is delivered by system, they mainly called as an effector. So effector means toxin, the same things. Effector and toxin is the same word basically. So they also produce on antitoxin or immunity protein, the same operon. So if this toxin is produced, at the same time, the immunity protein is also produced. And this immunity protein can binds or binds or somehow prevents the toxic activity of the toxin in the attacker cell. But this only toxin is delivered by the system to the neighboring bacteria, not the immunity protein. Immunity protein remains inside. And this toxin is delivered to the neighboring cell, and this toxin kills the neighboring bacteria and it gets killed. But bacteria cannot differentiate between competitor bacteria and its own sister cell. So it also delivers toxin to its own sister cell also. But this own sister cell has the antitoxins. Even this toxin is delivered by the system to the own sister cell. Because of the presence of the antitoxin, it don't get killed. So only the competitor bacteria gets killed, not his own sister cell. In our lab, we're mainly working on Vibrio parahemolyticus. So Vibrio parahemolyticus is as a model. So it's a gram-negative. It's mainly found in marine bacterium and it's a major cause of seafood-borne gastroenteritis. So it's a has pathogenic role. It can and it also have Type VI secretion system, and we also have antibacterial activity. And this is all the different gene which is required for the Type VI activity. Okay. So nowadays is a antibacterial. Nowadays we use basically antibiotics to kill two different pathogenic bacteria. But the antibiotic resistance is a major concern now in health industry because there's a lot of bacteria is coming which is becoming resistant to a lot of antibiotics. So there is a different approach also needed to solve this problem. As you can see, this Type VI secretion system has the ability to kill bacteria, naturally kill bacteria. So we can use this system to target the bacteria also. But we can't use Vibrio parahemolyticus as a model. So in our lab, I'm basically working in two different aspects of this system. If we can establish this Type VI secretion system in a good bacteria, because all bacteria are not bad, and then you can choose whatever toxin I want to deliver, then you can target the pathogenic bacteria and you can kill the bacteria. Okay, so basically this is two parts and I am working on the establish the system into the non-pathogenic bacterium is a good bacterium, and also that finding the new toxin which can be delivered by the Type VI. So today I will talk only about the second part, that there are toxin or effectors delivered by the system to find the new effectors or new toxin which can be delivered by the system. So there is several methods to find out new toxin which is delivered by the type 6. I'm not discussing all the methods that today I'm discussing only the method I use. I use some bioinformatics approach to find new toxin. What do you do? Like this is the this TIP protein basically. This TIP protein is this called VGRG or maybe VGRG protein. So this is mainly conserved in throughout the Vibrio parametrica's different strain. So this protein is very conserved. We look for this VGRG protein in different strains of Vibrio parahemolyticus. And we found on 3 gene operon like this one on 4470, 4465, on 4460, which has the which could have predicted the toxin which is delivered by the type 6. There are several criteria to check that. So we thought this could be because this 2 gene operon is mainly found in the downstream of the VGRG. And we also found a previous report found that the effectors are mainly present at the neighborhood of the VGRG gene. So we thought this could be the potential effector or toxin which is delivered by the type 6. So we tested this effector and immunity, whether using different method. And we also found this operon in the Vibrio parahemolyticus strain which are found in this region. Basically, Vibrio parahemolyticus is mainly found all over the world, but we found this operon and Vibrio parahemolyticus strain from isolated from this region. Okay. So we tested. We wanted to test whether this is really the effector immunity which is delivered by type 6 or not. So I'm not showing the exact result. I'm telling you that cartoon so that to understand how it works. So we do bacterial shape competition assay. So let's think about if we use wild type as an attacker and wild type as a prey. What will be that? So in presence of type 6, if toxin is delivered by type 6 to the neighboring cell in wild type against wild type, because wild type both has effector, both has immunity, and this effector is delivered by type 6 to the neighboring cell, but it has the immunity. So it should not be able to kill wild type against wild type. But if we use deletion of effector immunity as a prey, so in the effector immunity there is no effector and no immunity in the prey cell. So during attacking from the attacker cell, the toxin will be delivered by type 6 to the neighboring cell, but there is no immunity to protect the bacteria, so the prey will die. So that indicates this is the probable effect toxin and antitoxin system. But how we can confirm that this is delivered by type 6, not by other one? So what we do, we use attacker as a type 6 negative strain. Type 6 negative strain means we delete one of the essential component of type 6. That as I showed you earlier, there's a lot of genes are required for the type 6 activity. If we muted one of them, type 6 is not working. So I use one of the deletion strain as an attacker. So there is no type 6. So toxin will not be able to deliver by type 6 to neighboring cell. So there is no killing. That means this is the up to this we can say this is the effector and immunity. And this effector is delivered by type 6, not by other system. Then we need to know that which one is the effector. So then if we delete the effector, basically one of the genes, so if there is no effector, so there will be no toxin, so there will be no delivery, so no killing, so prey will survive. And next thing, which is the immunity gene. Then if you use the immunity, use wild type against delta effector immunity, should be able to kill because you deliver effector to the neighboring gene. But if we express the immunity gene from a plasmid, so there will be a lot of immunity. So even effector is delivered by type 6 to the neighboring bacteria, because there is a lot of immunity, it should be able to protect against the toxin. So there will be no killing. Using all this strategy, we find out that this 14465 is the toxin and 14460 is the antitoxin, is the immunity gene which protect from bacteria. Okay, then now we know that this is the toxin which is delivered by type 6. But how? How is carried out the toxic activity? Okay, to know that, we clone the toxin under a promoter, inducible promoter. This is P-ARA. So P-ARA means in absence of arabinos, there will be no transcription, so no toxin expression. But in presence of arabinos, this will be induced that there will be not induced transcription, or there will be no toxin activation. So before going to the main result, I just want to show you that how that strategy I used. First, as a negative control, we express a non-toxin, which is not reported as a toxin. What will be there? So in presence of no arabinose, there will be very low concentration of non-toxin, almost nothing, or that's you can see like 1 copy. So, and in presence of arabinose, there will be a lot of expression of the protein, but it's not toxic to the bacteria. So there will be not toxin, bacteria will be healthier. But if we clone the toxin, so there is no toxin, there will be very low concentration of the toxin, but still can be toxic to the bacteria. So you can say partially toxic, but in presence of arabinose, there will be a lot of toxins. So you'll see bacteria getting killed, bacteria is sick. Theoretical. As you can see here. Yes, this is the repressed condition, or maybe there is a no arabinose condition. So is a non-toxin, which if is non-toxin, is grows healthier here as well as here. Sorry, here and there. So this is, if you compare these two, so non-toxin is grows similarly in presence and absence of arabinose. But if you use the toxin here, so it's not, there's a very low copy number, very low expression of the toxin is also toxic to the bacteria. So it's not growing healthier like the non-toxin one compared to this one. And in presence of arabinose, there's no point question of growing. So now you can know from this result we can say the expression of the toxin have the deleterious effect on the bacteria. So it's bacteria has a toxic effect on the bacteria. Now we wanted to know this full protein is from 1 to 449 amino acid. That what's the region is required? This full protein is required, or some region is to depend the toxin activity. So we made all the deletion, all the different truncation form of this protein. Like I can see to 4.9, here we took this one from 95 to 4.9, all different truncation. And we streak in presence and absence of arabinose. And we found that the C-terminal region, like from 283 to 4.9, this is the required and essential for its toxic activity. So C-terminal domain of the protein is responsible for its toxic activity, and this is 283 to 4.9. This is sufficient. And we name this domain as pony. I will tell you later how we name it pony, but for now you can remember we name this domain as a pony. Okay, so pony has a toxic activity on E. coli. So when we look for pony domain containing protein, we found this is found in all the different bacteria. It's found almost near about 1500 others bacteria also contains this pony containing protein from gram negative to gram positive. Okay, and when we analyze all these 1500 proteins, whose pony containing proteins, we found a motif basically. So bigger is the letter, that means more conserved. We found a conserved motif. So like we found this is KX3GE motif. That's you can say this E is like highly conserved, like D is highly conserved, G and I is a highly conserved motif. And from Barnes prediction, we found that this motif in sometimes is found and may have some nucleus activity. So we wanted to test that basically. To test the nucleus activity, we need to purify the protein. We need a purified protein to check that. So we took the advantage of affinity chromatography. What is affinity chromatography? So during cloning, we put some codon at the C-terminal end of the toxin, like which represent the his amino acid. So, like I think CSE is represent the codon for histidine. So you can add 6 CSE, CSE, CSE. So there will be 6 histidine at that C-terminal end of the toxin. And we overexpress the toxin and pass this cell lysate through a column, through a nickel NTA column. What will be there? So let's say you want to, this is red one is your desired protein. So in the cell lysate, there will be 1000 proteins. So only that protein which contain his tag will bind here, and there is some non-specific binding too. And then we'll wash it to remove all the unwanted proteins. So all the unwanted protein will be removed during washing. And then you can elute it, basically using the elution buffer. So this is only the same buffer. This is dependent on the imidazole concentration. If we increase the imidazole concentration, this is the same, washing to elution. And we purified all three protein, like from 1 to 4.9, the full length protein, and 283 to 4.9, as well as we found purify 1 to 4.9, which is mutation, there's one mutation in the aspartic acid. We change the aspartic acid at 335 position to alanine. Because in the previous result, we found that if we mutate that aspartic acid 335 to alanine, it lost its toxicity. It's not toxic. So that's a negative control to that. And when we took the purified protein and incubate, because in the bioinformatics, it's predicted that it could have some nucleus activity. So we incubate the purified protein with genomic DNA, with isolated genomic DNA. And I took DNase1 as our positive control. In our experiment, whatever in the lab we do, we took always do two experiment, two control. One is positive control, one is negative control. So here we took positive control, the DNase1, which have DNase activity basically. And also as a negative control, we took BSA, that is bovine serum albumin, which has no DNase activity, not reported. As you can see, and DNase activity is when you use DNase1, you can chew up the DNA, even if it's a magnesium dependent or independent way. But in BSA, it can't degrade. And when we incubate our genomic DNA with our purified protein, that full length protein, that's 1 to 4 to 9, it can degrade the DNA. But the degradation depends upon the magnesium chloride, because the magnesium chloride is act as a cofactor. It's required magnesium chloride for the degradation. And also, if you use only C-terminal length, like from 283 to 449, it can also degrade the DNA. So the C-terminal end of this protein has the DNase activity, it can degrade DNA. As you can see, the point mutant one, that you see the D335A, where the aspartic acid is mutated to alanine. When you mutated the aspartic acid to alanine, it's lost its DNase activity. Okay, so pony possesses DNase activity. Then again, we did some bioinformatics analysis. When we analyzed the pony domain containing protein, and when we look the N-terminal domain, we found that pony containing domain also carries a several domain at the N-terminus region. Like basically, in some cases, it also contains some protein LXZ, because LXZ domain. This LXZ domain is mainly required for the delivery by type 7 sequence system. That means, as you can see, pony is delivered by different sequence system, not only type 6, depends upon the N-terminal region of this protein. Like this is a toxin domain, it's a delivery domain, it's a ball and socket type of things. So if it's delivery domain, if we change delivery domain by type 6, it can be delivered by type 6. If we change to type 7, like here LXZ, it can be delivered by type 7. Like if we change to, so let's say DU637, then it could be delivered by type 5. So this type of toxin they call polymorphic toxin. So from there we call this is a nucleus activity. So domain, so that's why you call pony, polymorphic nucleus effector. So from there we name this domain as a pony. So as I showed earlier. So this pony is it can be delivered by different secretion system depending upon the delivery. So pony is delivered by different system. Now next question is that I told you earlier that this 14460 is the immunity gene. It can protect from bacteria from its self-intoxication or same toxic activity. But how? So again, we went back to E. coli. The principle is that if we express only toxin, so if the repressed condition is a few copies of toxin, that will be partially toxic. As if we overexpress it, we find a lot of toxin, so it will be completely toxic. As you can see here, if we express only effector. But if we express the toxin in presence of immunity gene, in presence of antitoxin, somehow antitoxin prevent that toxic activity. As you can see here, there in case of effector and immunity, in presence of araminos, though both the toxin and antitoxin are expressing, but not showing any toxin activity. It's growing, it's healthier basically. And these two is for OPAR. OPAR is a non-toxic gene, so it's grow earlier. So as you can see, it grows healthier. Now, next question is that this immunity protein, this is the immunity protein, can protect the toxic activity only this gene, or it can protect any toxin. So we took another toxin like 1263. This is completely different toxin. It also have DNase activity, but it's a completely different toxin. We found that this pony cannot prevent the toxic activity mediated by this 1263. As you can see, it's not growing here. So This 14460 can prevent the toxin's activity mediated by 14465. It's only against this toxin, not any toxin and any other toxin. But how? It can prevent, but it's how it's preventing. It's through direct interaction or through indirect interaction, I don't know. So there is one method to check the direct bacterial interaction is called bacterial 2 hybrid. So they call bacterial 2 hybrid system. The principle of the bacterial 2 hybrid system is that there is two domain, and the T25 and T18. I think this two domain is found in one protein found in Bordetella bacteria. If these two domains interact each other, then it can catalyze from ATP to cyclic AMP, and this cyclic AMP can activate that the promoter. So if you clone some reporter gene under this promoter, so you get the blue color basically. You clone there's a legacy, so everything happening inside the E. coli. So there you can see the blue color. Let's say you want to study the interaction of between X and Y. So what we do? We generally fuse the X protein with one of the thing, T25, and Y as a T18. If these two protein interacts, then there will be ATP to cyclic AMP. We will see the blue color. So we took all these different constructs and we want to study all these three different constructs with the immunity gene or pony I. As you can see here, when you took the full length protein from 1 to 449 and that immunity gene, it will see that blue color. So that means the full length protein can interact with the immunity gene. As you can see, blue color. But when you took the only N-terminal region, this N-terminal part, this N-T means N-terminal part, it is not interacting with immunity. As you can see, it's white. But if you do the C-terminal part, from 280 to 4.9, it can interact with the immunity gene, as you can see the blue color. So that indicates that pony I, basically immunity gene, interacts with the full-length protein as it's directly interact with the toxin domain, not with the N-terminal part. We use as a VAS6 as a negative control. So as I told earlier, we always use some negative control also. So as you can see, there's no interaction with VAS6, some random protein is not interacting. Okay, so there is another method also to study this interaction. So that's they call pull down assay. What the pull down means is how basically it's also based on the affinity chromatography. So let's say this is a nickel anti-resin, this is the history, and you want to study the interaction between the protein between this protein and another other protein. So it will be there. So you can express both the protein inside bacteria and add some nickel anti-resin. So nickel anti-resin will bind your bait protein, and this is the three protein. Let's say is interacting with this one. So if you wash it, all other protein will remove. Only this three protein will be there, and you can detect this protein, prey protein, using different method. It's called Western blot. Okay, so like this is represent this plot. This is the input. In input, I found all these three construct, like this one, the full length, and this is C-terminal, this is N-terminal, this is the point mutant, and this is the all are immunity genes. So here, all three construct and immunity genes are there. But in the output, the protein which is only interact with this toxin will be there. So as you can see here, when I use the full length protein, immunity protein is there. When I use only C-terminal length, this immunity protein is there. But when you use the N-terminal part, only this, there is no immunity protein. Again, the point mutants is interacting. So that means it's also confirming our previous result that immunity protein only interact with the full length protein and the C-terminal length. Now, this is the toxin, this is 1 to 449, and C-terminal is producing the toxin activity, and it's sufficient for toxin activity. Then what's the role of the N-terminal domain, what it is doing? We name this domain as a fix. I'll tell you again later how we name it as a fix. So what we found, like what's the role of the fix? If we remember our previous slide, if we use wild type as an attacker and delta effector immunity as a prey, so in the delta effector immunity, there is no immunity gene. If you use the full length toxin, so it will be delivered by the toxin to the neighboring cell and it will get killed. But if we delete the N-terminal region, the fixed domain, we found that there is no killing. So that indicates this toxin is somehow not delivered for the neighboring cell. So that means this N-terminal part is required for the delivery of the toxin, for the loading of the toxin. Okay, and we also again we do bioinformatics analysis using the N-terminal domain, using the fixed domain, and we found this fix is also found in more than 2,000 different protein, and it's only found in gram-negative. And we want to analyze this 2,000 protein. Again, we found a conserved motif here there. Okay, this is the conserved motif, and for now, this is first time is found in its conserved motif. So we named it as a fix because found in type 6 effector. So that's we name it fix. And we also found that this when we analyze this fixed domain, fix containing protein, again we going back, fix containing protein. And if we analyze the C-terminal domain of the fixed containing domain, and then we found the different toxic domain at the C-terminal of the fixed containing protein. And also found that only 25% bacteria have has the type 6 secretion system. Okay, but we found that almost the 96% of the protein which has fix also have type 6. So somehow fix is genetically associated with type 6, and fix is mainly found in the neighborhood of the one of the structural component of the type 6. So there is again, this is you can say the fix can be acted as a term delivery domain. So it's a delivery domain, like if we, whatever the toxin you want, you can fuse the fix, like all this is all are different toxin. If you fix as a delivery domain, you can change it basically like a ball and socket. So I'm done almost. So in summary, we found a new toxin effector immunity in Vibrio parahemolytic 12 to 97B, where this toxin is composed of two domains, this N-terminal domain and C-terminal domain. C-terminal domain is mainly responsible for toxic activity, whereas this immunity gene, it can directly interact with the toxin domain and prevent the bacterial self-intoxication. And then this toxin is mainly loaded in the type C secretion system and by fixed domain, because fix is mainly interacting with that system, not the toxin domain, not the pony, and is delivered by the type 6 into neighboring bacterium, and is delivered there. It's mediated the toxic activity. This pony mediated the toxic activity, basically degrade the genomic DNA, and it's get killed. So in Alexander, I would like to my supervisor, Dor, and this Muskis, he also work with me in this project. And Jasmine, he's the master student, as a lab, King as a lab manager, and Daniel is a PhD student, and also Aaron from another university, who also took part in this project, and also the funding and the ERC and ISA. And thank you for the listening. If you have any question, please.
Thank you, Professor. I'm just a student. If any student have any question or doubt, so please voice up.
Am I audible, sir?
Yes.
Yes, sir. I'm Rishabh. Sir, you said that type 6 secretion system is a character of only gram-negative bacteria.
Yes,
So,
yes.
my question is there any specificity of the cell in which the toxin is being injected? It also be a gram-negative bacteria, or it might be any other bacteria?
No, basically, but gram negative is a good question. Gram negative, this is a mainly used in bacterial competition between gram negative bacteria. So far, we know that it can deliver toxin only in gram negative bacteria. And sometimes I did not talk about that. It can also deliver toxin into that into eukaryotic cell, virulent cell, basically. But the toxin is different. Today I mainly talk about the toxin which is in antibacterial competition, basically, which can target only bacteria. But the same system can deliver the toxin into eukaryotic cells, so it can also have virulence effect. And last, I think 2 years back, there is another thing. It can target fungus also. But so far, there is no report that it can target any gram positive.
Okay, sir. Thank you, sir.
Any questions?
Sir? Sir, am I audible?
Yes, yes.
Sir, we are the students of microbiology. So, What are the emerging fields in near future we need to more study?
Oh, emerging field. Like there's a lot of bioinformatics so far I know. In bioinformatics, a lot of guys are working on microbiome project, basically gut microbiome. What are different type of microbiota, and there's something a lot of guys are working on CRISPR also. I think this year they got Nobel Prize who discovered CRISPR, and these two fields are maybe emerging for now. Synthetic biology.
Thank you, sir. I have another question.
Yeah.
Now we found many many bacteria which are resistant to many antibiotics. So
Yes.
how we can found new antibiotics which can kill these antibiotic resistance bacteria?
Okay, so this is one approach I am using, so it is completely different like antibiotics, but people also screen for antibiotics. So there's a different screening method to find new antibiotics. So different extract they use, so different things. So this is completely different. So this is one of the alternative approach I am taking, using the bacteria already use it to kill another bacteria. So if you can control it, and if you somehow establish in some good bacterium, so let's say E. coli, which you can eat. So that's one approach. But discovery of antibiotic is completely new, so a completely different field. So you can some isolate screening method to do that.
Thank you, sir.
Thank you.
Anybody? This
Hello,
Yeah.
the faculty of government. I have, it was wonderful talk. I have really, it will help our student also. You said that in place of antibiotic, some bacteria are antibiotic
Yeah.
resistant. So use this system in place of antibiotic.
Yeah.
Because of some resistance, it can manifest some problem. So
Yeah.
can you say actually how can we use practically, how can we use this type 6 secretion system in place of antibiotic? Practically, that means
Okay.
there
But
may be some
actually,
idea.
this is a emerging field. This is in field still work is going on, so I can't say in that way. But the idea behind is that, let's say if you use this type 6 system, we know that this is can deliver the toxin to the neighboring bacterium. Okay. So if we can establish this type 6 in the bacterium, let's say what we eat basically, like in E. coli, which we add as a probiotics basically. So let's say if you can establish this system in one of the probiotic strain, like in E. coli, and there is a lot of study, you can regulate the system also. In our lab, I'm also working on the regulation, that like you can know that you can regulate when you want to activate it, when you don't want to activate it. So then if we can eat the bacteria, so it should be able to kill all the pathogenic bacteria which are resistant to antibiotics basically. That's the idea basically. And also nowadays a lot of papers comes with last year I think. There it can find, and it's also contact dependent. So you need to contact with the bacteria. So there is some they call nanobody expression. So they can, this particularly you can target the particular bacteria also using the same system. But all are in vivo, all are in tube. In the laboratory system, no one knows what will be in the in vivo, basically in human or something like that. That study it still is going on. Work is going on. Is it clear?
Okay, and okay, one more.
Yeah.
Some bacteria that is the same species that is secreting the type 4 secretion system. That we can use the type
Yeah.
4
The thing is that
system.
yeah, I understand. But Yeah, that's the thing is that look, different the bacteria can even the different same strain. Let's say Vibrio parahemolyticus, I work on let's say one strain. It can also kill the different strain. It can only don't kill the same strain. Okay, only the sister cell, because each bacteria has a different effector. Each bacteria has a effector immunity repertoire. So it will not kill only the same strain, but it can all kill the separate strain. And also you can manipulate it. Basically, you can choose if you find a lot of a toxin. So you can change it basically. So you can change all the different toxins. So okay, you can target this one. You can change it. So you can play with that.
So the what toxin type 6 secretion system, so we can
Yeah.
secretion system. So we can
No, you don't need to change.
system.
Yeah, exactly. I'm saying that you can use the system. What does the system do? System only deliver toxin. So you can change the toxin basically. If you know, let's say 4 or 5 different toxin which deliver the same system, you can manipulate the toxin. That okay, A toxin is not working. A, B, C, D. You can put all 4 toxin together. So that 4 toxin can kill this bacteria. I'm trying to say that because
Okay, basically
it can be divided in 2 parts. One, yeah, you're right. So this is one is the toxin delivery, which is delivery apparatus. Another one is which delivers toxin. So this is 2 parts. So you can play with the toxin, not with the system.
okay. Thank you.
Thank you.
Sir, am I audible?
Yeah.
Sir, pathogenic bacteria identification process through in vitro and in vivo methods.
Pathogenic. Sorry, I don't understand.
Pathogenic bacteria identification process through in vitro and in vivo methods.
No, pathogenic bacteria in vitro, in vivo. Look, some bacteria is pathogenic, like say Vibrio cholera. So you can identify by in vitro also by doing sequencing. So if you do say, suppose you isolate the DNA, you can get for sequencing. There this is the Vibrio cholera, this particular strain, which will be pathogenic. But if you want to confirm, you have to do the in vivo. So you need to do I don't do that, but in the several lab they infect with the eukaryotic cell with that. Yeah. Or mouse model, using some models mouse to infect with the pathogenic bacteria to see it's dying or not.
Thank you, sir.
Okay.
We reached the examination of the session now. I would like to invite the coordinator, Dr.
Sir. Okay. Now, with this, we come to the concluding moment of today's webinar. On behalf of our college, on behalf of our department, and also on behalf of our all faculty members, on behalf of all students, I extend a very heartfelt gratitude to Sir Dr. Vishwana Jana, sir, who spared his time from his busy schedule to grace this occasion to deliver this talk. It is a very wonderful talk, very informative talk. And I also thankful to him, sir, for very wonderful interaction. Today we had an opportunity to hear about his thoughts. This will surely be encourage our student in their future progress, and we will hope that he will definitely enlighten us again. And after the pandemic is over, on behalf of our college, we invite you, sir, to please come to visit our college. And again, I thanks for his wonderful talk. Thank you, sir.
Thank you very much. One funny story. I wanted to be part of the Ramkrishna Mission Rohra. So after passed my higher secondary examination,
Okay, so you are all student. Our
no, I'm not student. I wanted to be.
okay.
Then, but my home is from Medinipur, so I'm very older, so I don't know that time that Kolkata and everything. So I was very late. So that time when I went there, they said no, admission is closed now. So it's
So
wonderful feeling.
now we invite you, sir. Please visit to our college. Please visit to our ashrama. We are
Thank
quite
you
well
very much.
invited.